Welcome guys to Learn Extra. Today we're doing grade 12 revision. So I'm actually going to ask Kathy, what are you guys going to do today?
No, before we do that, we have to go back to Indy visiting our little boy who's done so well.
Guys,
Isn't that amazing?
I hope you guys were paying attention during that insert.
And guys, he hasn't got a silver spoon in his mouth, okay? He's from a very rural area in the Free State. Listen, don't knock the Free State. I was born in the Free State, all right. But from the Free State, not a city boy, nothing like that. He just worked hard. And people, and this is on a serious note, what you put in is what you get out. It's the same. Ty, come on, you're old enough, but you're old enough to know about relationships as well.
Exactly.
What you put in is what you get out. Work, what you put in is what you get out. So you want to have a happy life and all the rest of it. You know what? The only way you can do that is with money. And the only way you can get money besides disrespecting people and their human rights is to work hard. So work hard, people. There's no excuse not to get where you want to go. So, but I mean, like this, it's amazing.
He's awesome, guys. He got 5 distinctions. He's actually now got a bursary to go study at UCT doing chemical engineering, guys. No, that's one of the toughest degrees out there. So, guys, as Kathy is saying here, make sure you guys work hard, pay attention in class, make sure you're making notes and you're writing down all the stuff because it is important, and we want you guys to get those sky-high marks at the end of the year.
You know what? Just a little bit of work. Exactly. A little bit of work every day in every subject, and I promise you now, you are going to, you won't even begin to understand or comprehend the difference in the amount of hours you have to put into study. It's not the hours, it's the quality of what you do in those hours. You can't sit and listen to your cell phone and the radio and sort of watch a movie on your computer on the side here while you're busy studying. Shut yourself down, focus, learn for half an hour, rather than 3 hours of nonsense.
Okay, so guys, as you heard from Kathy and myself, make sure you got your pens and pads out, you Facebook me, talk to me. If you're having any issues, we'll cover that stuff. So guys, see you after this break. Welcome back, Mindsetters, to grade 12 revision today with Kathy. And guys, please pay attention to this woman because she's going to be teaching a lot of interesting stuff. And by the way, she actually just ran through like her all-star roster of people she actually just teach. You know, she taught Brian Abana, Graham Smith, and like a couple of other sports stars out there. So make sure you're paying attention because she knows what she's doing. So I'm going to hand over to Kathy and
Ty, I don't know if that's a very good ad because none of them became academics. They all went to play sport. But
they're educated.
Yeah, but that was King Edward's, you know. It's the boys there. A lot of the boys I taught, in the classes I taught, they sort of were at school to pass time to get to the sport fields in the afternoon. But you had to make sure that you taught them everything because you knew they weren't going to go and do it at home. So, or put very little work in. I love you all, my boys from Kes. Okay, we're going to be revising the grade 12 syllabus. People, Ty and I were also just talking about study methods. And, you know, maybe we're going to both speak to mindset and to Learn Extra and see if there isn't something we can put together on study techniques. Each person's different. I mean, if 10 of us went to breakfast, and Ty, I mean, like, you know, I'm going to come stand here with Ty. You know, Ty, like, if for example, you may like scrambled eggs with cream, and I may like scrambled or a poached egg. We all have different likes and dislikes,
but we're still
and
eating
things
egg.
that work for us, but we're still eating egg. So people, what we could do, and we'll speak to, I think we should to Learn Extra, and say, here are different study techniques. And studying is one thing, but you must be able to apply the stuff and link it. It doesn't help studying everything in little boxes. You don't live your life in little boxes, you know, so don't study in little boxes. And we need to do this with our learners. I think it's very important because then you can actually cut down on your learning time.
Exactly.
Quality
versus quantity.
And concentrating, cell phone off,
all nonsense aside, focused in a place where no one can disturb you, and you go for an hour and see how far you go.
Exactly.
All right. Yeah, I think it's much better. We need to do this. Hey?
Definitely.
All right, people. Term 1 revision. Okay, what did we do? We did DNA, we did RNA, we did meiosis, we did genetics, and we did genetic engineering. Okay, if you recall DNA and RNA, what are they? They are nucleic acids, all right? The DNA are hereditary characteristics. They sit in the nucleus of our cells, and that tells us who we are, what we are, where we are. And there are groups of people who believe that in that DNA we have almost like an inherited process where we have engendered knowledge of way back when. Something else that's very interesting is we have mitochondrial Eve, and what the guys have come up with and said that there are a whole group of people on this planet that have the same markers in their mitochondrial DNA, which can only mean that we are all sort of related in some other way, which is a little scary. But then you go to the Khoisan people, and they turned around and they said that in the two tribes, there were more variations in their DNA between the two tribes than there is between African people and European people. So, you know, and that's, these people live right next to each other. So, you know, there are all kinds of strange things that happen. All right, then we went through meiosis. Now remember, meiosis is for sexual reproduction, and if there's going to be sexual reproduction, what do we need? We need a haploid gamete plus another haploid gamete, so one from the male, one from the female, and the two haploid gametes will join during fertilization to form a diploid zygote. Right, so that was the whole meiosis process. What do you end up with? You end up with a diploid cell that undergoes reduction division, so it halves, haploid, haploid, and then it duplicates, very similar to mitosis, which is called meiosis 2, all right? And we end up with 4 unidentical daughter cells as compared to mitosis, which was for growth, repair, replacement, and reproduction in unicellular organisms, which we did with the grade 10s today. And mitosis, we end up with two identical daughter cells. Here we have 4 unidentical daughter cells. I'm just doing recaps here. Genetics, well, genetics, there were all those terms that we went through, and we spoke about chromosomes, and we spoke about DNA, and we spoke about alleles, and whether they are homozygous or heterozygous or dominant or recessive. We spoke about incomplete dominance, complete dominance, codominance, which is what we have in blood. So people, send me questions, please, because otherwise I'm just going to start at the beginning and try and work through as much as I can. Send those questions. Ty will stop me and he will ask me or tell me what it is you're sending, all right? So please, anything you're confused about, send. Okay, so here we're going on the structure of nucleic acids. This is a very, very nice exam question or test question. Where we can say, right, what does a nucleotide consist of? A nucleotide, or one nucleotide, will always have your phosphate ion, your pentose sugar, and look, I've even drawn it with 5 sides, and a nitrogenous base, and this is your protein part. Okay, that's your protein. Nitrogenous base, okay? And your phosphate and your pentose sugar form a backbone. So it'll be phosphate, and it'll go pentose sugar, and then it goes phosphate, and then down pentose sugar, and it forms like a backbone all the way down. And then attached to each phosphate, to each pentose sugar, you'll have your nitrogenous base. All right, now, if we look at the structure, we have our nitrogenous bases. You get 2 groups. You get your purines with a U and your pyrimidines with a Y. Okay. You get 2 types of purines and 2 types of, actually 3 types of pyrimidines, but we'll get to that. So your purines, you have adenine, okay, and guanine, so A and G. And your purines and your pyrimidines always bond together. You don't get 2 purines and you don't get 2 pyrimidines bonding with each other. All right? That's why they are called complementary bases. Nitrogenous bases are complementary bases. Okay, they're complementary. Purines always bond with pyrimidines. Always, always, always. Okay, so purines, adenine and guanine, so A and G. And your pyrimidines, look at this. Pyrimidine has a Y, cytosine has a Y, and thymine has a Y. So if they've got a Y, especially if they give you a multiple choice and say, which of the following is a pyrimidine? Well, pyrimidine's got a Y, so all you do is you look for cytosine or thymine, right? Your purines, adenine, guanine, they don't have the Y's in them. Okay, so cytosine and thymine, but remember this, adenine, which is your purine, always bonds with thymine, and guanine always bonds with cytosine. So A to T and G to C. Always. So before you answer any question on nucleic acids, in the top corner of your paper, you write A equals T, G equals C. So your cytosines bond with guanines, guanines with cytosines, thymines with adenine, adenine with thymine. That is if you have DNA. Okay, in RNA, nothing's bonding. So there are no weak hydrogen bonds. Remember, RNA is just a single strand. So with RNA, there is going to be no complementary bases, but what RNA will have is, instead of thymine, so there'll be no T's on any kind of RNA molecule, whether it's messenger, ribosomal, transfer, doesn't matter. If it's an RNA, there won't ever be a uracil, a T. There'll always be a U for uracil. So just think, pyrimidine's got the Y, cytosine's got the Y, thymine has the Y, and uracil is just the Y with a leg cut off, you see? So instead of thymine, on an RNA it will be uracil, and that's how you'll be able to tell if it's a DNA molecule or an RNA molecule. If it's DNA, there'll be thymine. If there's RNA, there'll only be U's and no T's. All right. And remember, pyrimidines always bond with purines. Why? Because they are complementary bases. Learn this. You should know it. I've gone over it enough times now. Okay, this is just a strand of DNA. Here you'll see phosphate sugar, pentose sugar, phosphate pentose sugar. There you'll have guanine to cytosine. Here you've got adenine. So on this side, you are going to have, let me just see if I've got the right pen here.
Kathy, actually, while we're on that, I actually have a quick question from Sinalo. She's dying to find this out. She wants to find out what's the difference between transcription and translation.
Okay, good question. Transcription, now, just listen to what the word means. To transcribe means to copy something over. Okay, in the old days, the monks used to take books. Remember, books were all handwritten, and they would transcribe this book and copy it as another book. And then those books would go to the kings and the queens and the lords and the ladies and the what's, because they're the only people who could read. In fact, only the men could read, not the women even, in the days I'm talking about. So to transcribe means to copy. So let's just get some to the bottom of this page here. Okay, so let's write this. Transcription. Okay, so transcribe is to copy. Copy exactly. Okay, so what we've got is a strand of DNA. So here's my DNA. I'm not going to draw in the phosphates and the pentose sugars, they're here. I'm not doing the detail. You'll see my little wiggle woggles here are supposed to be your phosphates. Okay, let me just get this away. I don't know why this is doing this. Stop. Okay, there we go. Okay, now here we're going to have our nitrogenous bases. Okay, there we go. This is a strand of DNA. But we want to see what's on that DNA. So what happens? The DNA in between here, it has weak hydrogen bonds. We have an enzyme that breaks that. So we end up with our DNA strand looking like this. Okay, so let's say that's adenine. So this would be, now remember, if that's adenine, that must be thymine. If this is guanine, that must be cytosine. If this is thymine, that must be adenine. And it's in groups of 3. Those groups of 3s are called codons. All right? Now, transcription, we are going to be copying, but what? We're copying our DNA. Because on that DNA is the information we need in order to make a new strand of DNA, or what we need to make something called proteins. Now remember, our proteins are very special to our body. Your proteins are not the same as anyone else on this planet unless you've got a twin brother or sister. Okay, so here we go. The DNA is now going to, I'm only going to be putting in the nitrogenous bases as the DNA opens up. So as it opens up, we're going to have thymine and adenine, and we're going to have guanine and cytosine, and adenine and thymine. So that's exactly that there. So along comes our messenger RNA. Now what does messenger RNA do? It acts as a little messenger. It comes, it transcribes, so it copies the information on the DNA, and it takes that to the ribosome, which is where proteins are made. Come on, you know that. You know that from grade 10. Why do we have ribosomes? They are for protein synthesis. Synthesis means to make. Right, so that's messenger RNA's job. It comes along and it says, okay, hold on, and it works in little groups of 3. So let's see, what's the opposite for T? Remember, A equals T and C equals G. That's all you need to remember. So we say, right, thymine, which means this must be an A, and this C, which means this is G, and this A, will remember in RNA, there is no thymine, only uracil. And it's only in RNA. So instead of thymine, we have uracil. That's my messenger RNA, so here it sits. And it's going to be adenine and guanine and uracil. Right? There, it has now transcribed the DNA. How? It's copied the complementary bases. So by copying the complementary bases and coming in as the complementary bases, it is going to be the opposite of what the DNA strand was, because it's the complementary base that's happened here. Okay, that's transcription. Now, we're going to go to translation. Now you know that the word translation means to take something that's in another language and put it across in your own language. Ty, how are we doing for time here?
We're good to go for an ad break if you want to.
I think, okay, let's go for an ad break and then we come back to translation. So I want this to sink in about transcription. Mm-hmm. So remember transcription? Okay, we've taken the complementary bases of the DNA. And that's our messenger RNA, because it's going to take that message to the ribosome. All right, people, remember that. Think about that. Over to you, Ty. Here we go.
So guys, I hope this is very helpful and is informing you guys. Make sure if you guys have any questions, if you're lost anywhere, again, post them to Facebook, and I'll get chatting to you, and we'll get Kathy to answer those questions that you're dying to find out about. But for now, we'll see you after this break. Welcome back, Mindsetters, to grade 12 revision, doing life science today with Kathy. So guys, again, I hope you're making notes. I hope you're writing this stuff down. Again, if you guys are lost anywhere, guys, go to the page, ask the questions, talk to me so I can talk to Kathy and we can get round to whatever questions that you guys are lost on. So I'm going to hand over to Kathy now.
Thanks, Ty. Kuni, thank you very much. I'm definitely going to hunt you down, hun, because I want some of those posters. All right, so, but as we go on here quickly, let's finish translation. So transcription, the messenger RNA is going to do the fit in and do the complementary bases to the information that's on that DNA. All right, complementary bases. No thymine, only uracil. All right, now, that little messenger RNA goes beady, 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 and it goes out through the little nuclear pore, and it goes ch-ch-ch-ch-ch-ch-ch, and all the way till it gets to the ribosome. And what is it doing? It's bringing its information. So along it comes here, let's do it like this, and we've got A and G and U. And what happens now is, let's get another color here. Along comes transfer RNA. And transfer RNA goes to fetch an amino acid, and it comes along, and it does this. It says, okay, well, hang on, if that is an A, that is A, and that's G, and that's U, then I'm going to be T, but remember, no thymine, so it's going to be a U, and a C, and an A. And then it goes, and it goes and picks up the amino acid. It then goes to the ribosomal RNA, little r, ribosomal RNA, who then takes that amino acid, and it takes it and it punts out this little amino acid and then another little amino acid until we have this whole chain of amino acids, which will then eventually make up one protein. Okay? But remember, this is your ribosome. So what is translation? It's when the messenger RNA goes to the ribosome and the transfer RNA then takes this information, all right, as the complementary of the information on the messenger RNA, and we now have the same as what was on the DNA. Just to show you this, and if you remember this and you learn this, you cannot go wrong. This is on the DNA. This is on the messenger RNA, and this is on the transfer RNA. So translation is to translate the information from the DNA. So here we go. Let's take our DNA strand, and we say the DNA strand that we're going to be looking at, we're going to only look at one triplet base or codon, and it's going to be, let's go GAT, GAT, all right? GAT. So our messenger RNA comes along and it says, okay, cool, there's my code on, because it codes onto the DNA, and it says, okay, so G would be C, A would be T, but remember it's RNA, so there's no T, so it's going to be uracil, and T would be A. There's my messenger, my messenger RNA. I want you to come down. There we are.
It's being like one of your students, rebellious.
Moving it around. Okay, so here, my messenger RNA is going to be, look at this, people, the opposite. So it's the complementary to the DNA. All right? That's my codon. Now it goes chup, 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 and it gets to the ribosome. When it gets to the ribosome, the transfer RNA says, cool, check this out, and it is now the opposite to the messenger RNA. It's the complementary. So what are we going to have? The transfer RNA is going to be, what's the opposite to C? G. What's the opposite to U? Is A. What's the opposite to A? Remember, no thymine, it's going to be uracil, because it is still RNA. Now look here. This transfer RNA and the DNA are the same. There's G, there's A, and instead of thymine, we have uracil. Why? RNA, DNA. So these two are the same. Okay? And DNA and RNA are opposite, and messenger RNA and transfer RNA are opposite. And because these two are opposite, they end up being the same. That's all you have to do. If you understand what I've done here, you'll never, ever, ever, ever get anything under 100% for your nucleic acid questions.
And then Kathy, yeah, while you're on that subject, I think there was someone who wanted to quickly find out, in fact, not even quickly find out, if you could go in depth in terms of pedigree, the pedigree chart.
Pedigree chart. Okay, cool. Here we go. Hang on, let's just get our, I know there's a slide here. John put a pedigree chart on for me. I'm just quickly zipping here. Oh, here we go. Ha, look at this. Okay, people, if you get a pedigree tree or a pedigree chart, you listen, rub your hands in glee, because that is full marks. If you get anything under full marks for a question on a pedigree chart that you are given, geez, then I don't know. Then I really don't know, because it is so easy. What you're going to do is you're always going to look for the two recessives. Now, just to remind you, okay, if it is, okay, homozygous dominant, if the characteristic is homozygous dominant, it will always be written with 2 capital letters. So let's say for brown hair, or in this case, we're talking about people that can roll their tongues and people that can't roll their tongues. Okay? Rolling your tongue is dominant over not being able to roll your tongue. Or it may be the other way around. You never ever know what they're going to tell you. They could tell you someone with black hair is recessive to someone who's blonde hair. Blonde-haired, okay, you must read your questions. If they tell you that the recessive gene is written with a capital R, then you know that, and you know that it should be a capital letter means homozygous dominant, and a recessive gene would be a small letter. What they're doing is they're testing your ability to apply your knowledge. Read your questions. Very important exam technique. All right, so homozygous dominant would be 2 capital letters. So I'm going to put here X and X, okay? And homozygous recessive would be a capital, wait, hang on. A little x and a little x. And if it is heterozygous, then it's going to be a mixture, so it'll be capital and small. All right, so that's just to refresh your memories. Now, we went through, when I did this with you, there were only 4 different types of crosses, okay? And if you learnt those 4 crosses that I gave you, and I know that Indy put them up on the Facebook page for you, just learn those 4. Just learn those 4 crosses, because that's it. And 90% of the time they're going to give you 2 heterozygous crosses anyway. But here, what we are going to do is we're going to look at, they tell you that a female non-roller and a male non-roller, so the circles are females, the males are squares. Not always, they could swap it around, but for here, that's what they're showing you. If they blank, they can't roll. If they dark, they can roll. But now remember, if it is a dominant trait, we don't know if it is homozygous dominant. We don't know if it is homozygous dominant or heterozygous. Because if it is complete dominance, you are always going to have the dominant trait coming through. So we may look at someone and they may have black hair, but they could be capital B, or they could be capital B, small b, because the dominant gene is coming through. All right, people, it's only if there's incomplete dominance that you will have red and white makes pink, or black and white makes gray. If there is codominance, black and white would give you black with white spots like a Dalmatian, or white with black spots like a Dalmatian. Or blood group A plus blood group B gives you blood group AB. So there you've got codominance, both traits dominate equally. Right, just remember that or make sure you know the difference between that. But let's go to our pedigree chart. So we always are going to work backwards. We don't know what the parents are, but the fact that these two parents have a white, so you've got a black there and a black there, and they give you a white. And here we've got two whites that end up with a white baby here. But we take the white and the black and we cross them, we end up with a black. Just looking at that, if I've got two, and forget about tongue rolling, it doesn't matter what the trait is. If I've got two and they give me something different that comes through, then it means the following. And this is what you will write, and this is what you will look for in any pedigree chart. You're going to look for the two recessives. So let's say, okay, in this case it's tongue rolling. So we're going to go with a capital R. I just want to get the, okay, we're going to say a capital R means that they can roll their tongues, and a small r means that they can't. Okay, so this is going to be recessive, and the capital R is dominant. Now remember, heterozygous, we don't know whether they're heterozygous or homozygous, except if we do the following. You see G here? In order for mom and dad at A and B to have G, and G is little r, it means that both mom and dad must have a little r gamete. Because this one's got two little r's. One came from mother, one came from father. But they're dark, so therefore, or they can roll. So therefore, we say that they are heterozygous. They must be heterozygous because this little one goes there, that little one goes there. We do our cross here. Capital R, small r, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, small r, small r. This was your third example I gave you, or third type of cross that you get. Two heterozygouses gives you a homozygous offspring, but it's a homozygous recessive. Okay, and that would be a boy, because the squares are boys. Right, here, these two have a child. But now look here. If H and G have a baby and K is the result, K is small r, which means H has to be capital R, small r. Now, if H is capital R, small r, it means that one of his parents must have had the small gene. So, it's either capital R, or capital R, small r, or here, it is capital R, small r, or capital R, capital R. Okay? So if it was that way or that way, you end up with your baby here. How do we know it's capital R? It has to have a small r or a recessive gene. How? Because they produce, G and H produce K. And K is two recessive genes. It's homozygous recessive. Okay, now, here they are not shaded, which means they're non-rollers. And guess what? If there are only recessive genes, doesn't matter what you cross, you're always going to have a baby that's recessive. Okay, now, J has to have a recessive gene. Why? Because his father, G, is homozygous recessive. But if we look at him, look at her, she is shaded, so therefore she must be heterozygous. People, are you following? All you do is you go to all the recessive genes. They tell you here what's recessive. And you go and you write all the recessive, homozygous, recessive. Now, if these two parents, L and M, have kids that have the recessive traits, homozygous recessive, it means both of them must be heterozygous. Okay, and these two have a baby. Now that baby is shaded, therefore it has to have the dominant gene. These two join up, and the fact that they have babies that are recessive, they have to have the dominant gene. So that is what they call a pedigree chart. So if we look at this and we say, right,
here. Kathy, before you actually go on, because I think we might just need a little bit of an ad break so the guys can catch their breath.
Okay, cool. Let's have an ad break.
So guys, make sure you just go quickly grab a snack, but make sure you come back with your pens and pads ready to make notes. And guys, again, Facebook me at www.facebook.com/learnextra if you've got any issues or any questions or lost anywhere. So guys, we'll see you after this break. Welcome back, Mindsetters, to another lesson for Learn Extra Live. We're doing grade 12, Life Science with Kathy. She's doing some revision. So I'm actually going to let Kathy continue what she was doing in the last, before we went to break.
Are you following? Because I'm going to test you at the end of this.
Ooh, that makes this tricky.
Okay, people, steps when answering a pedigree chart. Okay, the first thing you do is you read the key. And from that key, you are going to find step 2. You're going to find the recessive trait, okay, or recessive characteristic, okay. And from there, you'll be able to see, is it the shaded one? Is it the unshaded one? Is it, unless you're doing a sex-linked type disease, like for example hemophilia or color blindness or short-sightedness, something like that, you are going to look at, you don't have to worry about male and female, you're going to look at just whether it's blank or whether it is shaded. All right, once you find out what the recessive trait is, you go and you mark all recessive traits. Okay, you go there and you say, okay, look here, all the little white circles or blank circles, they are all my recessive ones. So I go, and you go and write all the recessive ones in. From that, you can actually figure out what the parents are. Because if the offspring has 2 recessive genes, I mean, it stands to reason that, look here, if there's baby, and baby has a little r, well, hello, one came from mommy and one came from daddy. So, hello, means mommy and daddy must have one, especially if they show the dominant gene, which in this case was capital R, capital R. So they both can roll tongues, but they produce the baby that can't. It can only happen if they're 2 recessive genes. I hope that makes sense to you. Ty, any other questions?
Yes, there is one quick one. If you could please go into more in-depth with the whole complete, incomplete, and codominance.
Okay, cool. I'm just going to write because I haven't got time to find slides. Okay. Let's do this. Okay, I'm going to have to use white and yellow. Okay, so here we go. We say, right, for complete dominance. Okay, this is Mendel's first law, which says that when there are dominant characteristics, and it's complete dominance, okay, and I mean there's all the fancy wording, but this is basically it, that when you have parents that cross with opposite and opposing dominant and recessive genes, when they cross, you're going to end up with the dominant characteristic showing in all the babies. Okay, so you've got your complete dominance, means that whatever that characteristic is, whether it's black, whether it's brown, what, if I take black plus white, the offspring are going to look black. Okay, so capital B and small b, and I'm going to end up with offspring that are capital B, small b. Why? It's complete dominance. Right, the dominant color will always, or trait will always come through. Then, so that one's easy. Right, then we have incomplete dominance. Now, it's not complete. Complete, that dominant gene goes, ha, I'm going to dominate over the recessive gene completely. All right, the only time the recessive gene will come through is if there are two and it becomes homozygous recessive. But here, incomplete dominance says, you know what, let's have a partnership here, hey? So we've got red plus white will be pink. Okay, or black plus white equals grey. Or they'll take cattle and say brown or red cattle and white cattle, give you rowan cattle, because rowan's like an in-between color. It's like that's sort of a reddish, gingery color, okay? Incomplete. So they're saying, right, you've got capital R plus small r, and you end up with capital R, small r. So in the heterozygous state, there's a sharing, there's a compromise. It's incomplete, there's a compromise. It's like partnership should be, all right? Black plus white, and we end up with gray. So in the heterozygous state, you'll see a mixture, a compromise of the two. And then we get codominance. And with codominance, now codominance is sort of, okay, you know what, you have your side of the room and I will have my side of the room, but we'll share the room. You see where with incomplete dominance, it's saying, you know what, you can use my desk and I'll use your chair, and we sort of share. But with codominance, it's we happen to be in the same room, there's the line. You have your half, I'll have my half, and we will share it equally. So what you've got with codominance is if I take red plus white, okay, I'm going to end up with red plus white spots or markings. Okay, if I have a black cow and I cross that black cow with a white cow, I end up with a cow that looks like a Friesland cow, which is a cow with black with white spots, or the other way around. Your Dalmatians, that's what a Dalmatian is, okay? It's your black and your white, and it says, let's share. So what are our genotype? Would be capital R, small r, and here I'm going to have capital R and small r where they share equally. Right, that's your codominance. Now, with animals and plants and all the rest of it, what we work with, where you are concerned in your syllabus, and this is just to make you understand because I've used the same colors all the way through, is that here we look at blood and we say, right, what have we got? How many different blood groups do we have? We're looking at codominance. We have blood group A, and that's actually AO. We have A, which is AA. we have B, which is actually BO. And we have B, which is actually BB. Okay, this is the trait that we see. This is what's in the genes. All right, and then we have A and B, which is AB. That's our universal acceptor or recipient. And then we have O. And O is OO. Why? Because it is recessive. So you've got to have 2 OO genes. It's called multiple genes. It's called, so here we're working with multiple genes for multiple characteristics, which is A, B, and O. So here, O is recessive, so it doesn't matter. 2 O's are going to give us O blood. And it's your most common blood, and it's the one that can be donated to anyone. Okay? Here, you've got, it's either your dominant gene and your recessive, which means that recessive goes. And that's your, that is your characteristic. Or it's AA, and you end up with A. B is either BO, and it completely dominates the O, or BB. And if we have an A plus a B, that's what we get there. And people, when you look at blood groups, remember there are 3 genes that determine your blood group. One is A, one is O, and one is B. And O is recessive. So A, mother, and a B, father, and they have a baby. That baby can either be, now look at this. And this is why we say that blood groups can only prove who a father is not. They can't prove who the father is. Why? Because there are actually 12 blood groups. There's 6 groupings, and then you've got rhesus positive and rhesus negative. So then ultimately there are actually 12. Now, if I want to prove someone's the father, you're going to have a problem because, hello, take all the people in this world and divide them by 12, and that's pretty much your blood groups. Okay, it's like the star signs. But you can say someone isn't. So if a father is AB and a child is O, that kid doesn't belong to that person. So you can cut him out. But if that child is AB and the father is A or B, hello, he could be the father, but so could a whole bunch of other people. All right, so we do it like this, and we say, if we have A, if the mom is A, and the dad is B, okay, the child, if the mother is, okay, and it also says if the mother is AO, or AA, and BB, or the dad could be BO, and the mother could be AA, and the mother could be AO, and the father could be BB. Now look at all the combinations here. If we say these 2, all right, and you end up with the A and the A, that child could be AB, that child could be AO, and the child could be BO. So pretty much the kid here could be, oh, hang on, and the child could be OO. So with this combination, you can end up with any child. Here, guess what? That kid can only be AB. Here, if we look at this one here, you could end up with a child who is AO. You could end up with a child that is AB, and that's it. And here, you've got the same kind of combinations as you have there. So the child can either be A, B, or the child can be B, O. So people, if I tell you the parents, the mother is homozygous A, the father is homozygous B, and the child is O, you can sue the hospital because you can say, no, 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 this baby is OO, not mine. Dad's homozygous A, mom's homozygous B. We can't have a child that has a blood group that wasn't even part of our genes. So you see, you can only prove what isn't. You can't prove that the child is yours. All right. Possibilities increase, however. Okay, any other questions?
We do have a couple. One, Basil wanted to find out if you could just go over genetic disorders and sex-linked characteristics.
Sure. That's going to... How much time have we got? I've got 3 minutes.
3 minutes.
Okay. All you've got to remember is, Okay, very quickly. Okay. The mother is going to be XX, because remember, we have gonosomes, somatic cells, and then we have our sex cells or gonosomes. Now, with the sex cells, in the female, it's XX, and in the male, it's X, and I've just lost. No, man, come. And the male is X, Y. Now, this Y chromosome is actually a short-legged X chromosome. All right? It looks like that. Okay, if the female is carrying something, a trait, and she's carrying that trait over here, there is no allele over here in the male to compensate it. All right, whether this is dominant, well, it's going to dominate, or if it's recessive. Oh, my foot is cramping terribly. If it's recessive, it doesn't matter. The gene is there. There is no allele on this side. Sorry. There is no allele to compensate. So whatever this characteristic is will be seen in the poor male. Because if it crosses, this is your crossing. X, and I'm doing this very simply, and the Y, and we'll take our red and we'll put it here. Okay, now we've got X, 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 Y, X, Y, because we've got 50% chance that it's boys, 50% chance that it's girls. But now look at this. This characteristic there, okay, is going to sit there, and it's going to sit here. So which means that this daughter here becomes a carrier. All right, this one is normal. This boy here is normal, and this one here is going to be affected. So hemophilia, colorblindness, short-sightedness, all kinds of sex-linked diseases, genetic diseases stuck here. Why? Because this poor little thing ends up with that X chromosome that's infected. All right, Ty.
Yes. So that brings us,
sure,
after such a long day.
It's because it's a holiday.
Exactly. It just feels so much longer. So guys, I hope this was helpful. I hope you guys were paying attention, wrote down notes. Guys, awesome stuff on the Facebook page. I just want to say thank you to Kuni, Kyla for posting, Kashifa for helping out. Guys, you guys are awesome. Always a pleasure to have you guys. And guys, again, keep posting, keep posting, keep posting. Make sure you write on the page. And from us at Mindset Learn, we're saying exactly. And we'll be back on the 16th with math. Bye.
Cheers.